Viruses. Viruses are obligate intracellular pathogens. Obligate intracellular pathogens are pathogens which cannot reproduce outside their host cell. Their reproduction is completely dependent on intracellular resources. They use the machinery and metabolism of a host cell to multiply. Structurally, virus possesses a protein coat called capsid. Capsid is made up of structural units called capsomeres. Capsid enclose nucleic acid, either DNA or RNA, but never both. Virus particle may be enveloped or naked, that is without envelope. Envelope may show projections called spikes. Spikes help viruses to get attached to host cell. There may be tegument, which is a cluster of proteins that lines the space between the envelope and capsid. The life cycle of a virus begins with attachment to a host cell. Attachment involves specific binding of viral capsid or envelope proteins to specific receptors on the host cell surface. After attachment with the host cell, the virus enters into the cell by membrane fusion or receptor-mediated endocytosis. After this, viral capsid is removed by viral enzymes or host enzymes or simply by dissociation, and the viral gene is released into the host cell. This is followed by the transcription or translation of the viral genome that leads to the de novo synthesis of viral genome and proteins. For some RNA viruses, this stage may involve reverse transcription, that is the synthesis of DNA from RNA. After de novo synthesis of viral genome and proteins, the newly replicated viral genome gets enclosed into viral proteins, that is capsid, to form new virions that are ready for release from the host cell. A virion is an intact virus particle. The newly formed virions are released from the cell by sudden breaking of the cell membrane or budding through the membrane to form virus envelope. These released virions are now ready to infect the new host cells. Antiviral drugs. Antiviral drugs are used to treat viral infections. Most viral infections resolve spontaneously in an individual having a normal immune response. Antiviral drugs act by arresting the viral life cycle at various stages such as virus attachment to host cell receptors, penetration, or uncoating. Uncoating is the release of viral genome in the host cell, inhibiting virus-associated enzymes such as reverse transcriptase, DNA polymerase, etc., inhibiting viral transcription and translation, interfering with viral regulatory proteins, interfering with glycosylation, phosphorylation, sulfation, and so on, interfering with the assembly and release of virions. Classification of antiviral drugs. The first classification is based on viruses against which a drug acts. This classification includes drugs act against herpes viruses, drugs act against influenza viruses, drugs act against hepatitis viruses, and drugs act against retrovirus, that is HIV. Classification of antiviral drugs according to mechanism of action. It includes agents inhibiting virus attachment, penetration, and early viral replication. Next group is neuraminidase inhibitors. Neuraminidase is found on the surface of influenza viruses that enables the virus to be released from the host cell. Next is agents interfering with viral nucleic acid replication. Fourth group is agents affecting translation by the ribosomes. Fifth group is antiretroviral agents, that is anti-HIV. This group include reverse transcriptase inhibitors, protease inhibitors, entry fusion inhibitor, CCR5 receptor inhibitor, and integrase inhibitor. Chemical classification of antiviral agents. It includes purine nucleoside analog, then pyrimidine nucleoside analog. Next is adamantine amines, then thiosemicarbazide, peptide analogs, thiazole carboxamide, sialic acid mimetic, sialic acids of cell surface glycoproteins and glycolipids are the receptors for many viruses such as influenza virus, coronavirus, etc. Then interferons and miscellaneous.